So today's lecture will be on the lymphatic system, the T lymphocytes, and the structures of the lymphatic system. So at first, when we're talking about the lymphatic system, there are three main types of lymphocytes in the human body. There's the T lymphocytes, the B lymphocytes, and the NK cells, or otherwise known as the natural killer cells. Understanding that all three migrate through the lymphatic system in search of an antigen or an expression of an antigen. The T lymphocytes at first are going to make up roughly about 70 to 85% of the body's total amount of lymphocytes. They express a plasma membrane called a coreceptor, or a CD4 coreceptor, that recognizes a particular antigen. So antigens are located on any type of foreign body or pathogen that enters into the human body. For example, the helper T lymphocyte has a CD4 coreceptor located on its plasma membrane here, otherwise known as a T4 cells. The T4 cell's job is to initiate and oversee the immune response by first presenting, as the picture shows here. So presenting the antigen for other lymphocytes. So the CD4 coreceptor will connect into the antigen of the foreign body for presentation to other lymphocytes. Or what they do is, after they've made connection to the antigen, the CD4 coreceptor makes connection to the antigen, it'll secrete a hormone called a cytokine, which is located here, these. So it'll go through a series of mitosis, secreting this hormone cytokine. These meiotic divisions will produce more helper T lymphocytes, but they also help stimulate or allow for other lymphocytes in the human body, such as the B lymphocytes, which have the antibodies, or the cytotoxic T lymphocyte, which tends to destroy or kill the pathogen, and it encourages other formations such as macrophages, which the job of a macrophage is to engulf and present the antigen on its plasma membrane. So the first type that we'll look at, other than the helper T lymphocyte, will be the cytotoxic. Their coreceptor, or T8 cell, is located here. So the coreceptor D8 coreceptor, or otherwise known as a T8. Their job is to come in direct contact with an infected or foreign cell. So here we have the antigen present on the infected cell. The CD8 coreceptor will bind to that. And then once it binds to it, it will then come in direct contact and kill the cell. These only activate after it's been activated by the T lymphocytes. So when we go back here, so these cytotoxic T lymphocytes are only activated with the ability for the mitosis of the T lymphocyte, helper T lymphocyte, and the cytokine release, which allows them to migrate into the area that's been affected. Eventually what we see is that the cytotoxic T lymphocyte will then detach from the infected cell, and then eventually we see the dead cell, or the infected cell, becoming dead or infected. So this diagram is going to show us a little bit more about the T lymphocytes. Probably the most important aspect is the fact that B lymphocytes, they have about 15 to 30% of the body's total lymphocytes, but they also contain antigen receptors to only one antigen and produce antibodies. So the most important aspect that we see is the helper T lymphocyte, as mentioned earlier, will go through mitosis, secrete the hormone cytokine to recruit more lymphocytes. But once it does that, then it recruits the B lymphocyte, which has the antibody located on its plasma membrane or surface. So once a B lymphocyte has been recruited or activated, um, most B lymphocytes are going to divide into plasma cells. So the first thing that we see once the antigen is presented to the B lymphocyte, if that antigen is compatible with the B lymphocyte, they'll connect. We'll see that the mitosis will produce plasma cells, and the plasma cells' most important aspect is that it's going to produce antibodies. So once it produces antibodies, these antibodies can be short-lived. They can last for a week or up to a year. But also during the process of mitosis, what we see is the division into a memory B lymphocyte. These memory B lymphocyte respond to re-exposure to the particular antigen, and they are already equipped with the antibodies. So once we see that, they're called a memory B lymphocyte. These B lymphocytes can have immunity to the antigen for years or a lifetime. If the antigen is present again, then we don't have to go through the initial steps. This B lymphocyte will then divide into more plasma cells with more secreted antibodies, and then they'll divide again into more memory B lymphocytes, which then increases the immunity to that particular pathogen. So during this process, we have to remember that all of your lymphocytes originally start in your red bone marrow. So this process is called lymphopoiesis. It's a process of lymphocyte development. So here we have a picture of a red bone marrow. And the first thing that we see is it's eventually all these lymphocytes are going to originate in the red bone marrow, but their maturation can be in different sites. So in this example, what we see is with lymphopoiesis, we'll see the division from a hemopoietic stem cell to a lymphoid cell. And this division, we can see migration to other organs for maturation, such as the thymus, or it can stay in the red bone marrow, divide into a lymphoid stem cell, and then divide and mature into a natural killer cell or a B lymphocyte. All of these eventually will travel into the blood and enter some form of a lymphatic structure, such as the thymus, the spleen, the lymph. So in this example, in the red bone marrow, the maturation would be occurring in the red bone marrow, dividing into a natural killer or B lymphocyte, eventually going into the cardiovascular system or blood flow, or the lymphoid cell can migrate to the thymus, where we see the lymphoid cell develop or differentiate into T lymphocyte. And again, they are going to then migrate into migrate and travel through the blood to the particular structure for maturation. So some of the lymphatic nodules that we see in the human body, in particular, the first one we'll start with is the lymphatic nodule called the tonsils. The tonsils have a discontinuous membrane which has an outer edge called a crypt. The crypt allows for the material to facilitate its identification and then allow for substances to enter into the medulla area of the tonsil. So we have a couple of different tonsils. The first one would be the pharyngeal tonsil. These are adenoids. They're also called adenoids. They're located in the posterior surface or posterior wall of the nasopharynx. So the nasopharynx would be kind of the division between the nose and the pharynx or the pharyngeal aspect of the mouth. The next one we see is the palatine tonsils. These are located mostly in the oral cavity, and they're on the posterior lateral region of the body. And then the last one we see here is the lingual tonsils. They're located in the posterior 1/3 surface of the tongue. Again, their job is to trap foreign particles to allow its identification for lymphocytes to come in and to destroy, depending on what type of antigen is present. So your thymus has a couple of main functions. Its main function is T lymphocyte differentiation and maturation. It's a bilobed organelle, so you can see here it has 2 lobes. It's located superficially to the heart, and it's consisted of these lobules that are fused together. It has an inner or an outer shell called, in each lobule, it has an outer shell called a cortex and an inner shell called a medulla. So inside of the cortex, we'll see immature T lymphocytes. So these are going to usually be immature T lymphocytes. So we'll just call that immature T lymphocytes. And in the medulla, we see mature T lymphocytes. The thalamus is going to continue to grow until puberty and then regresses in size and function in adults. In adulthood, T lymphocytes can only be produced by cellular division, not by maturation of new cells in the thymus. So in the slide previous, we saw the lymphoid cell coming into the thymus and developing maturation for the T lymphocyte. This would occur all the way up until puberty, and then after that we would see, as previously mentioned, the division of the T lymphocytes for immune function. The next structure that we have is called a lymph node. Its primary job is to filter antigens from lymph and initiate immune responses. So we see interstitial fluid being collected in the capillary beds. It's going to have an afferent pathway into a nodule or an afferent lymphatic vessel. Once it enters into the lymph node, it'll go through a series of sinuses. So it'll allow itself to either travel through the medulla, through the medullary sinus, or through the cortex, through the cortical sinus. So a sinus is just essentially a pathway to allow the lymph to filter through the medulla and the cortex. And each one of these are important because they're going to house different types of lymphocytes or macrophages. So the first thing that we look at is the capsule. So the capsule is surrounded by a tough connective tissue, and its internal extensions are called a trabeculae. They project into the nodules. It sort of compartmentalizes, so you can see the trabeculae here. It compartmentalizes here, and it's going to compartmentalize the cortex and the medulla. The lymph again, as mentioned, divides into the cortex and the inner medulla. They both have some lymphatic sinuses. And the first one we're going to look at is the importance of the medulla with the macrophages. So the macrophages, its importance is that it's going to engulf any type of pathogen. When we look at the lymphatic nodule here, we see that inside of these germinal centers, so inside of the germinal centers, we have the T lymphocytes and the inside of the germinal centers, we mostly have the B lymphocytes or any type of the macrophages. The T lymphocytes are going to actually be located outside of the germinal centers. So here would be kind of the line. Anything located inside would be considered to be inside of the germinal centers, such as the B lymphocytes or the macrophages, and anything along the outer edge, such as the T lymphocytes. So as the lymph is flowing through, as mentioned earlier, we have afferent pathways coming in. Inside of these afferent pathways, we have a series of valves. And once the lymph is filtered in, we're going to see it leave out through an efferent pathway. So basically, all we're looking for through this whole process is to make sure that any antigens that are present that are inside of the lymph, we could have recruitment of T lymphocytes, B lymphocytes, or engulfing through macrophages. So just a simple way of filtering the lymph before it enters back into the cardiovascular system. So the spleen has a couple of functions. Its job is to phagocytosize or use phagocytosis to destroy bacteria, old erythrocytes, red blood cells, and platelets, and to initiate an immune response if an antigen is present inside of the blood. So what we see is, first off, is the location. It's the largest lymphatic organ in the body. It's just lateral to the left kidney and lies close to the diaphragmatic surface. The splenic artery and vein here, they enter and leave the spleen through the hilum or the medial surface, just another name for the hilum is the medial surface. So these splenic arteries and veins are going to become important in multiple aspects for filtering the blood or the ability to bring in damaged red blood cells or erythrocytes into the spleen. So when we look at the function of the spleen, it's surrounded by a dense connective tissue called a capsule. Again, it has trabeculi. So here we see the outer edge or surface of the spleen is called a capsule. And then we see the trabeculi, which is just an extension of the capsule going in. The trabeculi are important because this is where we see a couple of different aspects. The trabeculi are important because we see it gives a pathway for the splenic vein to run into. So we can see how the splenic vein runs into the trabeculi or the cortex. So with that, what we see is different divisions. So the first one we have is the white pulp, and the other one is the red pulp. So all white pulp, as we see here, this whole region here, it's being innervated by the splenic artery, and all the red pulp, which is the outer surface, is going to be innervated by the splenic vein. Both of them have different types of functions. So starting with the outer edge, the red pulp, it serves as a reservoir for healthy erythrocytes and platelets. It can use phagocytosis to destroy old and defective platelets. The white pulp inside, their job is to as the blood flow is entering into the spleen is to initiate any immune response when any antigens or any antigen is present in the blood. So these different regions have important functions with maintaining uh, antigen-free blood. So that concludes the lymphatic immune response.